Beneath the ocean surface exists a vibrant and complex world teeming with life. These intricate underwater cities, known as coral reefs, are hotspots for biodiversity, providing habitat for countless marine species and supporting fisheries around the globe. Acting as natural barriers along coastlines, they reduce wave energy, protecting beaches from erosion and keeping coastal communities safe. But these fragile ecosystems on which humanity depends are disappearing, struggling against forces we can no longer ignore. While their decline is undeniable, there is hope. Through scientific innovation, we can still take action to restore and protect these precious ecosystems.
Hi there, I'm Nick Jones. I'm a research scientist at Nova Southeastern University, where I focus primarily on community dynamics on coral reefs so to see how they're changing and then why they're changing. Now, much of my research really is looked in Florida and looking at the kind of situation that coral communities are facing within Florida. And one of the primary things we've been seeing is due to the reduction in the number of colonies around due to disease, bleaching, multiple stressors really, which are leading to the degradation of reefs, is that we're finding isolation within populations or isolated colonies. If you have colonies which are isolated spatially, the chance of the gametes actually meeting is pretty low. So one of the things we've been looking at Nova Southeastern University is what techniques we can employ to try and promote sexual reproduction and get around this problem of isolation.
So I managed the Spawning Hub project since its inception in 2020. We established the spawning hubs to address the issue of reproductive isolation on our reef tract. So this was an opportunity to transplant conspecific species into these two spawning hub sites where they can effectively reproduce. In April and May of this year, 2024, we went out approximately 10 days after the full moon. We take out a team of 5 that consists of a captain and then 4 divers. The coral that we decided to launch for spawning is called the larvae. Its common name is the groove green coral. It's a really cool thermotypic broadcast spawner.
It creates habitat and structure on the reef and provides a home to many species of fish and invertebrates.
So once we get to the site, we hop in the water and we set up our spawning nets, which are mesh bags basically with a buoyant cone. And we can screw in these falcon tubes that collect the buoyant gametes when the colonies spawn. And once we see that they're done, we usually collect the tubes. We screw a top on, we write the tag number of what colony that these gametes came from, and we swim up to the surface and we hand them off to our fertilization crew.
The divers come up, they bring me the different falcon tubes from different colonies, so they're different genotypes, so I can put them all together at the right concentration. And because we presume at this point that the eggs have already been fertilized, so now they're already embryos, we actually want to get rid of the sperm. Because the eggs are buoyant, they'll be all at the surface, so you can get rid of the liquid that is underneath, that is the sperm. And then you just mix more water, and then you can use the gravy separator to get rid of the bottom part that has sperm. And then you have a clean culture, and you can put them in this conical tanks that have a recirculating sort of upwelling movement of water. So the labyrintiformis takes about 2 to 3 days to start having a larva that is swimming directionally and already looking for a place to settle. And they go into these trays where we have placed ceramic tiles that had been left in the same tanks as adult corals were for about 2 months. They have collected this microbial biofilm, this bacteria, and also there's might be some pieces of Cursus coralline algae that is usually a cue for this is a good place to settle. They will attach to it, and then they will go and undergo metamorphosis and become a little polyp.
So we grow these juvenile corals for about 9 months to a year in our land-based nursery, up to the size of a quarter, and only then send them back out to the ocean.
These lab-grown corals are eventually transplanted back onto Florida's reefs, where they will face the challenges of the modern oceans. With hope, they will thrive to adulthood, providing crucial habitat for marine life and contributing to the genetic diversity of future coral populations.